Hello, so today's chapter is chapter number 3, Plant Kingdom. In the previous chapter, we looked at the broad classification of living organisms under the system proposed by Whittaker in 1969, wherein he suggested the five kingdom classification, which is Monera, Protista, Fungi, Animalia, and Plantae. In this chapter, we will deal in detail with further classification within kingdom Plantae, popularly known as plant kingdom. We must trace here that our understanding of the plant kingdom has changed over time. Fungi and members of the Monera protista having cell walls have now been excluded from Plantae, though earlier classification placed them in the same kingdom. So, the cyanobacteria that are also referred to be blue-green algae are not algae anymore. In this chapter, we will describe algae, pteridophytes, bryophytes, gymnosperms, and angiosperms under Plantae. Let us give a look at classification within angiosperms to understand some of the concerns that influence the classification systems. The earlier systems of classification used only gross superficial morphological characters such as habit, color, number, and shape of leaves, etc. They were based mainly on vegetative characters or on the androecium structure system given by Linnaeus. Such systems were artificial. They separated the closely related species since they were based on new characteristics. Also, the artificial system gave equal weightage to vegetative and sexual characteristics. This is not acceptable since we know that often the vegetative characters are more easily affected by environment. As against this, natural classification system developed, which were based on natural affinities among the organisms and consider not only the external features but also internal features like ultrastructure, anatomy, embryology, and phytochemistry. Such a classification for flowering plants was given by George Bentham and Joseph Dalton Hooker. At present, phylogenetic classification system based on evolutionary relationships between the various organisms are acceptable. This assumes that organisms belonging to the same taxa have a common ancestor. We now use information from many other sources to help resolve difficulties in classification. This becomes more important when there are no supporting fossil evidence. Numerical taxonomy, which is now easily carried out using computers, is based on all observable characteristics. Number and codes are assigned to all the characters and the data are then processed. In this way, each character is given equal importance and at the same time hundreds of characters can be considered. Cytotaxonomy that are based on cytological information like chromosome number, structure, behavior, and chemotaxonomy that uses the chemical constituent of plant to resolve confusions are also used by taxonomic these days. 3.1 Algae. Algae are chlorophyll bearing simple thalloid autotrophic and largely aquatic, both freshwater and marine water organisms. They occur in variety of other habitats, moist stones, soil, and wood. Some of them also occur in association with fungi, which is lichen, and animals, that is on sloth bear. The form and size of algae is highly variable, ranging from colonial form like volvox and the filamentous form like eulothrix and spirogyra. A few of the marine forms such as kelps from massive plant bodies. The algae reproduce vegetative, asexual and sexual methods. Vegetative reproduction is by fragmentation. Each fragment develops into a thallus. Asexual reproduction is by production of different types of spores. The most common being the zoospore, they are flagellated and on germination give rise to new plants. Sexual reproduction takes place through fusion of two gametes. These gametes can be flagellated and similar in size as the eulothrix or non-flagellated, non-motile but similar in size as in Spirogyra. Such reproduction is called isogamous. Fusion of two gametes dissimilar in size as in species of Eudorina is termed as anisogamous. Fusion between one large non-motile static female gamete and a smaller motile male gamete is termed oogamous. For example, volvox and fucus. Algae are useful to man in various ways. At least a half of the total carbon dioxide fixation on Earth is carried out by algae through photosynthesis. Being photosynthetic, they increase the level of dissolved oxygen in their intermediate environment. They are paramount importance as primary producers of energy-rich compound which form the basis of food cycle of all aquatic animals. Many species of Porphyra, Laminaria, and Sargassum are among the 70 species of marine algae used as food. Certain marine brown and red algae produce large amount of hydrocolloids, which are water-holding substances. For example, algine, which is a brown algae, and carrageen, which is a red algae, which are used commercially. Agar, one of the commercial products obtained from Gelidium and Glaciaria, are used to grow microbes and in preparations of ice creams and jellies. Chlorella, a unicellular alga rich in protein, is used as food supplement even by space travelers. The algae are divided into three main classes: chlorophyceae, phaeophyceae, and rhodophyceae. 3.1.1 Chlorophyceae. The member of chlorophyceae are commonly called green algae. The plant body may be unicellular, colonial, or filamentous. They are usually grass green due to the dominance of pigment chlorophyll A and B. The pigments are localized in definite chloroplast. The chloroplast may be discoid, plate-like, reticulate, cup-shaped, spiral, or ribbon-shaped in different species. Most of the members have one or more storage bodies called pyrenoids located in the chloroplast. Pyrenoids contain protein besides starch. Some algae may store food as in the form of oil droplets. Green algae usually have a rigid cell wall made up of an inner layer of cellulose and outer layer of pectose. Vegetative reproduction usually takes place by fragmentation or by formation of different types of spores. Asexual reproduction is by flagellated zoospore produced in zoosporangia. The sexual reproduction shows considerable variation in the type and formation of sex cells, and it may be isogamous, anisogamous, or oogamous. Some commonly found green algae are Chlamydomonas, Volvox, Eulothrix, Spirogyra, and Cara. 3.1.2 Phaeophyceae. The member of Phaeophyceae or brown algae are found primarily in marine habitats. They show great variation in size and form. They range from simple branched filamentous form like Ectocarpus to profusely branched form are represented by kelps, which may reach a height of 100 meters. They possess chlorophyll A, C, carotenoids, and xanthophylls. They vary in color from olive green to various shades of brown depending upon the amount of xanthophyll pigment fucoxanthin present in them. Food is stored as complex carbohydrates which may be in the form of laminarin or mannitol. The vegetative cells have a cellulosic wall usually covered on the outside of a gelatinous coating of algine. The protoplast contains, in addition to plastids, a centrally located vacuole and nucleus. The plant body is usually attached to the substratum by a holdfast and has a stalk, the stipe, and leaf like photosynthetic organ, the frond. Vegetative reproduction takes place by fragmentation. Asexual reproduction in most of the brown algae is by biflagellated zoospore that are pear-shaped and have two unequal laterally attached flagella. Sexual reproduction may be isogamous, anisogamous, or oogamous. Union of gametes may take place in water or within the ooonium. The gametes are pyriform, pear-shaped, and bear two laterally attached flagella. The common forms are Ectocarpus, Dictyota, Laminaria, Sargassum, and Fucus. 3.1.3 Rhodophyceae. The member of Rhodophyceae are commonly called red algae because of the predominance of the red pigments are phycoerythrin in their body. Majority of red algae are marine with greater concentrations found in warmer areas. They occur in both well-lighted regions close to the surface of water and also at great depths in ocean where the relatively little light penetrates. The red thalli of most of the red algae are multicellular. Some of them have complex body organization. The food is stored as Floridian starch, which is very similar to amylopectin and glycogen in structure. The red algae usually reproduce vegetatively by fragmentation. They reproduce asexually by non-motile spores and sexually by non-motile gametes. Sexual reproduction is oogamous and accompanied by complex post-fertilization developments. The common members are Polysyphonia, Porphyra, Glacilaria, and Gelidium. Bryophytes 3.2. Bryophytes includes the various mosses and liverworts that are found commonly growing in moist, shaded areas as hills. Bryophytes are also called amphibians of plant kingdom because these plants can live in soil but are dependent on water for sexual reproduction. They usually occur in damp, humid, and shaded localities. They play an important role in plant succession or bare rocks soil. The plant body of bryophytes is more differentiated than that of algae. It is thallus-like, prostrate or erect, and attachment to the substratum by unicellular or multicellular rhizoids. They lack true roots, stem, or the leaves. They may possess root-like, leaf-like, or stem-like structures. The main plant body of bryophyte is haploid. It produces gamete, hence it's called gametophyte. The sex organ in bryophytes are multicellular. The male sex organ is called antheridium. They produce biflagellate antherozoids. The female sex organ is archegonium, is of flask shape and produce a single egg. The antherozoids are released into water where they come in contact with archegonium. An antherozoid fuses with the egg to produce zygote. Zygote do not undergo reduction division immediately. They produce a multicellular body called the sporophyte. The sporophyte is not free living but attached to the photosynthetic gametophyte and derives nourishment from it. Some of the cells of sporophyte undergo reduction division, that is meiosis, to produce haploid spores. The spores germinate to produce gametophyte. Bryophytes in general are of little economic importance, but some mosses provide food to herbaceous mammals, birds, and other animals. Species of sphagnum, a moss provide peat that have long been used as a fuel and as packing material for transshipment of living material because of their capacity to hold water. Mosses, along with lichens, are the first organisms to colonize rocks and hence are of great economical importance. They decompose rocks, making the substrate suitable for growth of higher plants. Since mosses form dense mats on the soil, they reduce the impact of falling rain and prevent soil erosion. The bryophytes are divided into liverworts and mosses. 3.2.1 Liverworts. The liverworts grow usually in moist, shady habitats such as bank of streams, marshy ground, damp soil, bark of trees, and deep in the woods. The plant body of liverworts is thalloid, for example, Markensia. The thallus is dorsiventral and closely appressed to the substrate. The leafy members have tiny leaf-like appendages in two rows on the stem-like structures. Asexual reproduction in liverworts take place by fragmentation of thalli or by the formation of specialized structures called gymi. In singular, gemma. Gymi are green, multicellular asexual buds which develop in small receptacles called gemmacops located in the thalli. The gymi become detached from the parent body and germinate to form new individuals. During sexual reproduction, male and female sex organs are produced either on the same or different thalli. The sporophyte is differentiated into foot, seta, and capsule. After meiosis, spores are produced within the capsule. The spores germinate to form free living gametophytes. 3.2.2 Mosses. The predominant stage of the life cycle of moss is the gametophyte, which consists of two stages. The first stage is the protonema stage, which develops directly from a spore. It is a creeping green branch and frequently filamentous stage. The second stage is a leafy stage, which develops from the secondary protonema as a lateral bud. They consist of upright, slender axils bearing spirally arranged leaves. They are attached to the soil through multicellular and branched rhizoids. This stage bears the sex organs. Vegetative reproduction in mosses is by fragmentation and budding in the secondary protonema. In sexual reproduction, the sex organs antheridia and archegonia are produced at the apex of leafy shoots. After fertilization, the zygote develops into a sporophyte consisting of a foot, seta, and capsule. The sporophyte in mosses is more elaborate than that in liverworts. The capsule contains spores. Spores are formed after meiosis. The mosses have an elaborate mechanism of spore dispersal. Common example of mosses are Funaria, Polytrichum, and Sphagnum. 3.3 Pteridophytes. The pteridophytes include horsetails and ferns. Pteridophytes are used for medicinal purposes and such as sound blenders. They are also frequently grown as ornamentals. Evolutionarily, they are the first terrestrial plant to possess vascular tissues, xylem and phloem. You shall study more about these tissues in chapter 6. The pteridophytes are found in cool, damp, and shady places, though some may flourish well in sandy soil conditions. You may recall that in bryophytes, the dominant phase of life cycle is gametophytic body. However, in pteridophytes, the main plant body is sporophyte, which is differentiated into true root, stems, and leaves. This organ possesses well differentiated vascular tissues. The leaves in Pteridophyta are small as Selaginella or large as in ferns. The sporophytes bear sporangia that are subtended by a leaf appendages called sporophyles. In some cases, sporophyles may be distinct compact structure called strobili or cones Selaginella equisetum. The sporangia produce spores by meiosis in spore mother cells. The spores germinate to give rise to inconspicuous, small but multicellular, free-living, mostly photosynthetic thalloid gametophytes called prothallus. These gametophytes require cool, damp, shady places to grow. Because of this specific restricted requirement and the need of water for fertilization, the spread of living pteridophytes is limited and restricted to narrow geographical regions. The gametophytes bear male and female sex organ called antheridia and archegonia respectively. Water is required for transfer of antherozoids, the male gamete released from the antheridia to mouth of archegonium. Fusion of male gamete with the egg present on the archegonium result in the formation of zygote. Zygote thereafter produces a multicellular well-differentiated sporophyte, which is dominant phase of the pteridophytes. In majority of the pteridophytes, all the spores are of similar kinds. Such plants are called homosporous. Genera like Selaginella and Salvinia, which produce two kinds of spores, macro and microspores, are known as heterosporous. The megaspores and microspores germinate and give rise to female and male gametophytes respectively. The female gametophytes is variable periods. The development of zygote into young embryo takes place within the female gametophytes. This event is a precursor to the seed habit considered as important step in evolution. The pteridophytes are further classified into four classes: Coelopsida, Lycopsida, Synopsida, and Pterospida. 3.4 Gymnosperms. The gymnosperms, gymno means naked and sperma means seed, are placed in the ovules and are not enclosed by any ovary wall and remain exposed, both before and after fertilization. The seeds are developed post fertilization are not covered. Gymnosperms includes medium-sized trees or tall trees and shrubs. One of the gymnosperm, the giant redwood tree Sequoia, is one of the tallest tree species. The roots are generally tap roots. Roots in some genera have fungal association in the form of mycorrhiza, pinus, while in some other, cycas, small specialized root called coralloid roots are associated with nitrogen-fixing cyanobacteria. The stem are unbranched cycas or branched pinus or cedrus. The leaves may be simple or compact. In cycas, the pinnate leaves persist for a few years. The leaves in gymnosperms are well adapted to withstand extremes of temperature, humidity, and wind. It conifers, the needle-like leaves reduces the surface area. Their thick cuticle and sunken stomata also help to reduce water loss. The gymnosperm are heterosporous. They produce haploid microspores and megaspores. The two kinds of spores are produced within sporangia that are borne on sporophyles which are arranged spirally along an axis to form lax or compact strobili or cones. The strobili bearing microsporophyles and microsporangia are called microsporangiate or male strobili. The microspores develop into a male gametophyte generation which is highly reduced and is confined only a limited number of cells. This reduces gametophyte is called pollen grain. The development of pollen grain takes place within the microsporangia. The cones bearing megasporophylls with ovules or megasporangia are called macrosporangiate or female strobili. The male or female cones or strobili may be born on the trees Pinus. However, a cycas male cone or megasporophylls are born on different trees. The megaspore mother cell is differentiated from one of the cells of the nucellus. The nucellus is protected by envelopes and composite structure is called ovule. The ovules are born on megasporophylls which may clustered to the form of female cones. The megaspore mother cell divides meiotically to form four megaspores. One of the megaspore enclosed within the megasporangium develops into multicellular female gametophyte that bears two or more archegonia or female sex organs. The multicellular female gametophyte is also retained with megasporangium. Unlike bryophytes and pteridophytes in gymnosperm, the male and the female gametophytes do not have an independent free living existence. They remain within the sporangia, retained on the sporophyll. The pollen grain is released from the microsporangium. They are carried in air currents and come in contact with the opening of ovules borne on megasporophylls. The pollen tube carrying the male gametes grows towards archegonia in the ovules and discharge their contents near the mouth of the archegonia. Following fertilization, zygote develops into an embryo and the ovules into seed. The seeds are not covered. 3.5 Angiosperm. Unlike the gymnosperm where the ovules are naked, in the angiosperms or flowering plant, the pollen grain and ovules are developed in special structures called flowers. In angiosperm, the seed are enclosed in fruits. In angiosperm are exceptionally large group of plant occurring in wide range of habitats. They range in size from small wolfia to tall trees eucalyptus. They provide us food, fodder, fuel, medicine, and several other commercially important products. They are divided into two classes, the dicotyledonous and the monocotyledonous. The dicotyledons are characterized by seeds having two cotyledons, reticulates venations in leaves, and tritamerous and pentamerous flowers that are having four or five members in each floral whorls. The monocotyledons, on the other hand, are characterized by single cotyledons seed parallel venation in the leaf and tremendous flowers having three members in each floral whorls. The male sex organ is in a flower in stamen. Each stamen consists of slender filament with an anther at the tip. Within the anthers, the pollen mother cell divide by meiosis to produce microspores which matures into pollen grains. The female sex organ is in a flower is a pistil. Pistil consists of smaller ovary as it is base, a long slender like stigma and style. Inside the ovary, ovules are present. Generally, each ovule has a megaspore mother cell that undergoes meiosis to form four haploid megaspores. Three of them degenerate and one divide to form the embryo sac. Each embryo sac has three-celled egg apparatus, one egg and two synagrids, three antipodal cells and two polar nuclei. The polar nuclei eventually fuse to produce a diploid secondary nucleus. Pollen grain after dispersal from the anthers are carried by wind or various other agencies to stigma of pistil. These are termed as pollination. The pollen grains germinate to the stigma and the resulting pollen tubes grow through the tissues of stigma and style and reach the ovule. The pollen tubes enter the embryo sac where two male gametes are discharged. One of the gametes fuses with the egg cell to form zygote. The other male gamete fuses with the diploid secondary nucleus to produce the triploid primary endosperm nucleus. Because of the occurrence of two fusions, that is syngamy and triple fusion, this event is termed as double fertilization, an event unique to angiosperms. The zygote develops into an embryo with one or two cotyledons and the PEN develops into endosperm, which provides nourishment to the developing embryo. The synergids and antipodals degenerate after fertilization. During these events, the ovule develop into seed and the ovaries develop into fruit. The life cycle of angiosperm is shown in figure 3.6. 3.6 Plant life cycle and alternation of generation. In plant, both haploid and diploid cell can divide by mitosis. This ability to leads to the formation of different plant bodies, that is haploid and diploid. The haploid plant body produces gametes by mitosis. This plant body represents as a gametophyte. Following fertilization, the zygote also divides by mitosis to produce a diploid sporophytic plant body. Haploid spores are produced by this plant body by meiosis. This is turned divide by mitosis to form a haploid plant body once again. Thus, during the life cycle of any sexually reproducing plant, there is an alternation of generation between gamete producing haploid gametophyte and spore producing diploid sporophyte. However, different plant groups as well as individuals representing them differ in the following patterns. First, sporophytic generation is represented only by one cell zygote. There are no free living sporophytes. Meiosis in the zygote result in the formation of haploid spores. The haploid spores divide mitotically and form the gametophyte. The dominant photosynthetic phase in such plant is the free living gametophyte. This kind of life cycle is termed as haplontic. Many algae such as volvox, Spirogyra and some species of Chlamydomonas represent this pattern. On the other extreme is the type wherein the diploid sporophyte is in the dominant photosynthetic independent phase of life. The gametophyte phase is represented by the single to few cell haploid gametophyte. This kind of life cycle is termed as diplontic. An alga fucus species represent this pattern. In addition, all seed-bearing plant, that is gymnosperm and angiosperm, follow this pattern with some variation wherein the gametophytic phase is few to multicelled. Third, bryophytes and pteridophytes interestingly exhibit an intermediate condition, haplodiplontic. Both phases are multicellular, however they differ in their dominant phases. A dominant independent photosynthetic thalloid or erect phase is represented by a haploid gametophyte and it alternates with the short-lived multicellular sporophyte, totally or partially dependent on the gametophyte for its anchorage and nutrition. All bryophytes represent this pattern. The diploid sporophyte is represented by a dominant independent photosynthetic vascular plant body. It alternates with multicellular saprophytic autotrophic independent but short-lived haploid gametophyte. Such a pattern is known as haplodiplontic life cycle. All pteridophytes exhibit this pattern. Interestingly, while most of the algal genera are haplontic, some of them such as Ectocarpus, Polysiphonia, Kelps are haplodiplontic. Fucus and Alga is a diplontic. Summary: Plant kingdom includes algae, bryophytes, pteridophytes, gymnosperms, and angiosperms. Algae are chlorophyll-bearing simple thalloid, autotrophic, and largely aquatic organisms depending on the type of pigment possessed and type of stored food. Algae are classified into three classes, namely chlorophyceae, phaeophyceae, and rhodophyceae. Algae usually reproduce vegetatively by fragmentation, asexually by formation of different types of spores, and sexually by formation of gametes which may show isogamy, anisogamy, or oogamy. Bryophytes are plants which can live in soil but are dependent of water for sexual reproduction. Their plant body is more differentiated than that of algae. It is thallus-like and prostrate or erect and attached to the substratum by rhizoids. They possess root-like, leaf-like, and stem-like structures. The bryophytes are divided into liverworts and mosses. The plant body of liverworts is thalloid and dorsiventral, whereas mosses have upright, slender axis bearing spirally arranged leaves. The main plant body of a bryophyte is gametophyte producing and is called archegonia. The male and female gametes produced fuse to form zygote, which produces a multicellular body called sporophyte. It produces haploid spores. The spores germinate to form gametophytes. In pteridophytes, The main plant is sporophyte, which is differentiated into true root, stem, leaves. These organs possess well-differentiated vascular tissue. The sporophytes bear sporangia, which produce spores. The spores germinate to form gametophyte, which require cool, damp places to grow. The gametophyte bear male and female sex organ called antheridia and archegonia respectively. Water is required for transfer of male gametes to archegonium, where zygote is formed after fertilization. The zygote produces a sporophyte. The gymnosperms are plants in which ovule are not enclosed by any ovary wall. After fertilization, the seed remain exposed and therefore this plant are called naked seed plant. The gymnosperm produce microspore and megaspore, which are produced in microsporangia and megasporangia borne on the sporophylls. The sporophylls, microsporophylls, and megasporophylls are arranged spirally on axis to form male and female cones respectively. The pollen grain germinates and pollen tube release the male gamete into the ovule where it effuses with the egg in archegonia. Following fertilization, the zygote develops into embryo and ovule into cysts. In angiosperm, the male sex organs, stamen, and female sex organs, pistil, are born in a flower. Each stamen consists of a filamentous and an anther. The anther produces pollen grains after meiosis. The pistil consists of an ovary enclosing only one to mainly ovules. Within the ovule is a female gametophyte or embryo sac which contains the egg cell. The pollen tube enters the embryo sac where two male gametes are discharged. One male gamete fuses with egg cell and other fuses with diploid secondary nucleus. This phenomenon of two fusion is called double fertilization and is unique to angiosperms. The angiosperm are divided into two classes, the dicotyledons and the monocotyledons. During the life cycle of any sexually reproducing plant, there are alternation of generation between gamete producing haploid gametophyte and spore producing diploid sporophyte. However, different plant groups as well as individuals may show different patterns of life cycle, haplontic, diplotic, or intermediate. Thank you.